So there is significant separation in the viral RNA copy number by treatment of this compound solidoside. As I said, it is very important to have good antiviral immunity. So how to test antiviral immunity, whether I am having the good antiviral immunity or not? So for testing the good innate immune sensor, or you can say the antiviral state, there is a protein known as RIG1. This is an RNA helicase which senses the viral RNA presence at a very early stage when the virus, the first virus or second virus comes inside the cell. So they recognize this RIG1 protein, recognize the presence of viral RNA and it starts secreting interferon alpha production. And as soon the interferon alpha production, the virus C, it almost gone down. So RIG1 protein we found that significantly enhances by the solidoside treatment at both mRNA level as well as the protein level. Now, interferon regulatory factor 3, which is in the downstream signaling cascade of this RIG1 and MDA5, we reported that IRF3, this is a protein which is responsible for enhancement in the type 1 interferon. So IRF3 production was significantly enhanced by this solidoside treatment in the viral infected cells. So it is clearly indicating that the solidoside is able to stimulate the antiviral state in our body. Similarly, it was further confirmed by another means because we had to test it in the different modern methods at the different transcriptional level, translational level, and other methods. And when you test it by different means, then you become confirmed that yes, it is not just a mere observation, but yes, it's actually causing the expression of increased interferon alpha. And we have tested it by different methods like translational and transcriptional approach, as well as by enzyme-linked immunosorbent assay, and we found that solidoside is very good in enhancing our antiviral cytokines. Other than antiviral cytokine, it is very important to inhibit the viral protein synthesis, and for that, there is a protein known as eukaryotic initiation factor 2 alpha. This phosphorylated EIF2 alpha inhibits the viral protein synthesis. It does not allow viral protein to go into the protein synthesis machinery. And therefore, this protein is very important to know whether this protein is upregulated by solidoside, yani rhodiola treatment or not. So we have treated the cells with the solidoside infected with the dengue virus and reported that the phosphorylation of EIF2 alpha protein is significantly enhanced. And this is clearly indicating, and because of enhancement in this phosphorylated EIF2 alpha, the virus protein expression we got suppressed. Now, as I said, cytotoxic T cells. There are two types of T cells. T cells are very important in the viral fighting against the virus immunity because we have two types of T cells. One is known as the helper T cell and the second one is known as the cytotoxic T cell. For killing the virus, cytotoxic T cells play important role, and this is a cytotoxic T cell which actually kills the virus. And if you can see, the solidoside treatment in the virus infected cell enhances the percentage of the cytotoxic T cells. Now, if I tell you how the cytotoxic T cell kills the virus infected cell, the virus, when infect a cell, it is processed inside our cytoplasm and its peptides are loaded on the surface of the MHC class 1. As soon it is loaded on MHC class 1, it is recognized by this TC cell or cytotoxic T cell. When cytotoxic T cells see the viral peptide presence, it goes near to that cell and make a channel between these two cells, the infected cell and the cytotoxic T cell. And through this channel, it pours a kind of protein, which is proteolytic in nature, and kills the infected cell by a means apoptotic death. It starts apoptosis in that infected cell and does not allow other cells to get infected. So apoptosis death is very clean way of killing the infected cell or cancerous cell by these cytotoxic T cells. Similarly, this is the natural killer cell. The virus is very clever. What it shades off all the MHC class 1 from the surface, host cell surface, so that cytotoxic T cell cannot see the presence of the viral peptides on the MHC class 1 and it can escape from the killing of the cytotoxic T cells. So we have another kind of cell which does not recognize the class 1 MHC associated viral antigens. It only recognizes those cells which do not express the MHC class 1. Like in case of tumorigenic cell or in case of the viral cells, infected cells, they does not express, do not express this MHC class 1. And therefore, it is the time of NK cells to get recognized these infected cell or tumorigenic cell and kill it immediately. So if your NK cells functioning is good enough, you will be protected from any kind of viral infection or any kind of, I can say, any kind of cancer, or you can say tumor genesis. And I can say that it is important that if we do pranayam regularly and yoga regularly, it is important to look into whether the regular yoga practice or pranayam practice enhances our NK cell functioning and cytotoxic T cell functioning or not. It is very important. And I think Morarji Desai Institute of Yoga should explore this type of research, because it will strengthen our fact that yes, not only the blood pressure, hypertension, and all those aspects, but it also maintains the good innate immunity or the cytotoxic T cells and NK cell functioning as well. And this is the treatment of the celluloid in the virus infected cell for this natural killer cell. And we reported here that the level of NK cells get upregulated. So in summary, I can say celluloid treatment strengthens our innate immunity, our NK cells, and cytotoxic T cell functioning. And therefore, it could be useful for managing the severe form of dengue fever and dengue shock syndrome. Now, third objective which I have evaluated is the role of papaya leaf extract in platelet augmentation and dengue virus in suppression. This is the Carica papaya leaves for the dengue virus treatment. Every season, if you see the dengue cases, people are just behind the papaya leaves. They will not get any papaya leaf in any papaya plant, because of all the people take out this papaya leaf. They don't know what could be the mechanism, but they think that this papaya leaf extract is very useful for treating the dengue fever. And we scientifically found that papaya leaf is, yes, usually very useful for the management of dengue fever. LCMS analysis we did for papaya leaf extract, and these are the compounds which we reported. These are different types of amino acids like L-serine, L-proline, L-cysteine, glutamic acid, tyrosine, and other flavonoids like catechin, quercetin, hesperidin, myristin, and carpane. Carpane is known to enhance the platelet count. We tested the different doses for safety aspect, and we found that the 100 and 200 microgram per ml of this papaya leaf extract does not cause any kind of cytotoxicity. So we further tested the virus infected cell which are treated with the papaya leaf extract, and we found that when the cells were treated with papaya leaf extract, the virus protein synthesis was significantly reduced or almost gone down. Then we tested whether papaya leaf extract is also able to enhance type 1 interferon or not. Because as I told you, the interferon alpha level is very important for a particular, for your immunity. If you are having good quantity of interferon alpha, you will not be infected with any kind of viral infection. And if you are suppose infected, it will be very mild, because interferon alpha is directly an antiviral cytokine, which can be stimulated by this. You can see the interferon alpha level was enhanced by papaya leaf extract treatment. We further interested in knowing whether it is causing any kind of toxicity to the RBC, because when you take any kind of plant-based drug, it is very important to look into the safety and toxicity aspect. And we found that RBC were not lysed. And I'm telling you one story of one plant. It was developed for the adjuvant. Adjuvant is a kind of compound which enhances the immunity when mixed with the vaccine grade antigens. So any kind of antigenic immune response can be enhanced by the adjuvant. And in one clinical trial, it was found that one adjuvant which was obtained from a plant that was causing RBC lysis, and that was leading to the hemoglobin, decreasing the hemoglobin. So it is very important safety aspect that you must be clear that it is not causing any kind of hemolysis when it goes into the body. We reported that in a very high dosage, the papaya leaf extract does not cause any kind of the RBC lysis. Now, most important aspect, whether papaya leaf extract is able to increase thrombocyte count, platelet count or not. This is because a scientific approach must be there to prove that modern medicine, through modern medicine approaches, traditional medicine can be accepted by everyone, whosoever is believing in either in the allopathic medicine system or any kind of medicine system. So this is one aspect that what we were looking, that is it really increasing the platelet count or not. So we deliberately decreases the platelet count by putting the cyclophosphamide drug to the rats, and in 2-3 days the platelet count decreased in the animal, and then we treated them with the papaya leaf extract. And what we found that yes, papaya leaf extract is significantly enhancing the thrombocyte count, platelet count. And you can see over here that in the 11th day it was significantly increased by the papaya leaf extract. So it is clearly indicate the reported observed that papaya leaf extract is very good in enhancing the thrombocyte count, even platelet count. I'm telling you one story. In case of cancer patients, in some patients chemotherapy induced thrombocytopenia occurs, and for those patients recombinant interleukin 11 treatment is given by the doctors, and this recombinant interleukin 11 is actually very expensive. One cannot afford the middle class income people cannot afford this type of treatment modality. So if papaya leaf extract is able to enhance the thrombocyte count, it could be one option for those who are actually in the need of the platelet augmentation. But remember, it is an inherent property of papaya leaf extract. It should not be taken by those who are healthy people, because it increases the platelet count. So if you take healthy people take papaya leaf extract, there could be a chances of enhancement in the platelet count, and increase in platelet above the normal may have deleterious effect, because it may make a thrombus, and it may cause the different types of stroke problems or cardiac arrest problems like that. So only those people should take it who are having the lower thrombocyte count. Now the question is, why papaya leaf extract able to increase platelet count? What does it do inside our blood, inside our body? So we were interested in knowing what kind of the factors responsible for enhancing the thrombocyte count. So we found that thrombopoietin and interleukin 6, these are the two important cytokines which actually increases the platelet number by putting the hematopoietic stem cell to the erythroid progenitor, then converting them to the megakaryocyte, which break down into different types of platelets. And remember, platelet is anucleated cell. It is a fragment of cell. So we found that papaya leaf extract enhances the interleukin 6 and thrombopoietin level. So I come to the conclusion of this aspect that these two plants, rhodiola, which is having the celluloside, an active ingredient or active compound, I can say, exhibits the anti-dengue activity by upregulating innate immunity, RIG1, MDF5, interferon alpha, phosphorylated EIF2 alpha, natural killer cells, cytotoxic T cells, and PLE exhibit anti-dengue virus activity and augment platelet count by increasing interleukin 6 and thrombopoietin expression. So PLE could be a potential antiviral agent against dengue virus infection, and also in case of those cases where the thrombocytopenia occurs. So until a remedy came, we should prevent mosquito breeding, use insecticides, use mosquito net, wear mosquito repellent, empty drain or cover all the things that can hold water, and plant some mosquito repelling flowers and herbs. There are some herbs which are actually mosquito repellent. These are like lemongrass, the marigold. All these plants can repel the mosquito. So with this, I came to the end of this session. Thank you all. Thank you for inviting me and having me on this weekly webinar. Thank you so much.
Thank you, sir. Thank you for such an elaborative and excellent lecture. सर, हमारे कमेंट सेक्शन में कुछ प्रश्न है, अगर आपकी अनुमति हो तो मैं कुछ प्रश्न लेना चाहूंगी.
Sure, ma'am.
सर किसी एक व्यक्ति ने कमेंट किया है कि जैसे आपने बताया कि पपाया लीफ खाने से प्लेटलेट काउंट में वृद्धि होती है, तो कोई ऐसी अन्य बीमारी आप बता सकें जिसमें हम अगर पपाया लीफ खाएं तो उसमें फायदा पहुंच सकता है?
जैसा कि मैंने बताया मैडम, पपाया लीफ एक्सट्रैक्ट थ्रोम्बोसाइटोपिनिया केसेस में लेना चाहिए, जहाँ पे मतलब प्लेटलेट डाउन होता है। तो प्लेटलेट डाउन कई तरह से हो सकता है, नॉट ओनली डेंगू वायरस causes the platelet decrease, but chemotherapy agent also, chemotherapy cases also reported to have lesser platelet count. Some drugs also causes the lesser platelet count. Even there are some autoimmune diseases, autoimmune problem where the thrombocyte counts are decreased. So drug-induced thrombocytopenia, chemotherapy-induced thrombocytopenia, or in case of the dengue or hemorrhagic fever also the thrombocytopenia cases are reported. So those cases could be considered.
Okay. Sir, thank you so much for joining us today and delivering such an excellent lecture. आपका बहुत बहुत धन्यवाद हमारा अनुरोध स्वीकार करने के लिए और हमारे दर्शकों को इतना सुंदर व्याख्यान देने के लिए। इसके साथ ही साथ मैं संस्थान के निदेशक महोदय डॉक्टर ईश्वर वी बसवरेड्डी सर का भी हार्दिक धन्यवाद ज्ञापित करती हूँ, जिनकी ऊर्जा और प्रेरणा से ही इस प्रकार के आयोजन को संभव बनाना हो पाता है। आपका भी बहुत बहुत धन्यवाद सर। साथ ही साथ संस्थान के संचार एवं प्रलेखन अधिकारी तथा हमारी तकनीकी टीम का भी हार्दिक धन्यवाद और हमारे साथ जुड़ने वाले आज सभी दर्शकों को भी हार्दिक धन्यवाद ज्ञापित करती हूँ। आप सभी को बहुत बहुत धन्यवाद। और संस्थान की परंपरा के अनुसार कार्यक्रम का समापन शांति पाठ के साथ। सर्वे भवन्तु सुखिनः, सर्वे सन्तु निरामया, सर्वे भद्राणि पश्यन्तु, मा कश्चित् दुःखभाग् भवेत्, मा कश्चित् दुःखभाग् भवेत्। ॐ शान्तिः शान्तिः शान्तिः। Thank you all.